Hi, my name is Cedra Ali, and welcome to my final project for Biology 105 Lab. For my project, I had to find a lab and publication that I was interested in, so I chose Dr. Steve Rogers, who specializes in cellular and developmental biology. Dr. Rogers has taught a number of courses, including my cellular biology course that I'm taking next semester, Biology 240. I found one of his publications, Microtubule Acetylation is Required for Mechanosensation in Drosophila, very interesting, and want to focus on it for my project. The researcher's hypothesis is that microtubule acetylation is required for touch in fruit flies. Hold on, I know this sounds complicated. It definitely was for me before I finished molecular biology, Biology 220, so let's break it down. Microtubules are tiny structures that are found in cells, and they play an important role in cellular movement and division. As for acetylation, I actually just learned what this meant in class very recently, so don't feel bad if you don't know it. Acetylation is the process of adding a tiny molecule, called an acetyl group, to a larger molecule, like a protein or a cell part. This change can affect how the molecule works and what it does inside a cell. Researchers in Dr. Steve Rogers' lab at UNC wanted to investigate if microtubule acetylation played a role in the sense of touch in fruit flies, which is what Drosophila means. The researchers did this using two different methods. First, they used a wild type or regular allele to identify what caused microtubule acetylation, which is Drosophila atubulin acetylase, shortened as DTAT. And they found that blocking this acetylation affects touch through observing the sensory neurons of the flies. Flies with less microtubule acetylation were less responsive. They then used calcium imaging to find why this was and found that the mutation of the DTAT increased gentle touch receptors. Despite how important calcium imaging was to discover the correlation between microtubule acetylation and fruit flies' sensory neurons, calcium imaging can do a lot more than just that. So what is calcium imaging? Calcium imaging is a process where scientists observe the changes in the amount of calcium in a cell. They use a fluorescent dye to make the calcium glow when the levels change. This makes it a lot clearer than attempting to inefficiently and inaccurately count calcium ions with just the human eye. It can be used to look at changes in sensory neurons like touch, hearing, smelling, tasting, and seeing. It can really be used in all types of fields that involve neurons like neuroscience, pharmacology, as well as medicine. So how does it work? The calcium indicator dyes consist of two parts. As you can see in this image, it's the site that bonds to the calcium and then the fluorescent light. By quantifying the changes in the calcium concentration, it can be represented by the intensity of fluorescence or how bright the light is emitted by a neuron. Some scientists further change the calcium indicator dyes to make sure they can easily cross the cell membrane, and if they don't do that, it has to actually be injected into the brain tissue. As shown, this is what's happening to the mouse in the image. If we were to actually complete the process in this graph, you would first have to prepare the dye you chose to attach to the calcium. You have to mix it with other solutions to ensure fluorescence or that it would glow, and you'd have to make sure to properly mix it. And then you'd place these neurons you collect from organism and cover neurons in the dark for the necessary time. For the mice in this experiment, it was 30 minutes, but it varies based on the organism. You then put the neurons at a microscope and collect an image at each wavelength through a camera. The change in calcium concentration is shown through the spikes in the graphs, and then they can be analyzed. Calcium imaging can be used to reveal how neurons process all kinds of signals, like pheromones, odors, touch, neurons in the spinal cord, muscle cells, immune cells, and much more. Thank you for watching. These are my references, and I hope you learned something.